Okay. All right. Thank you to ARM for inviting us to speak today, and I'll be giving an overview of our company, Fibrocell. We are a public company traded on NASDAQ, so I will be making some forward-looking statements today, and I'll refer you to the slide. So Fibrocell, if you're unfamiliar, is a gene therapy company that focuses on using autologous fibroblasts as a platform for targeting rare diseases in skin and connective tissue. And we have a lead program called FCX007 that is being developed for a disease known as recessive dystrophic epidermolysis bullosa. It's a rare blistering disorder that I'll give you some information on coming up shortly. We are in a phase 1 portion of a phase 1/2 study now, and we've just last week put out a top-line early data interim review, and I will talk obviously a little bit about that data on this presentation, and looking forward in the next coming quarter to performing some additional dosing and also moving into a pediatric population in the early quarter of next year. This development program has been granted orphan drug designation, rare pediatric disease designation, and fast track by the FDA, so we've worked very well with the agency developing this product. We also have 2 other indications. I won't be speaking in detail on these today, but we do have a program called FCX013 that is in preclinical analysis now. It's a genetically modified fibroblast that encodes a protein called MMP1 to target localized scleroderma, so an autoimmune sclerotic disease that can impede motion, can impede development, and we're working on trying to create a solution to help break up these sclerotic regions. There is an IND filing for this expected in the 4th quarter of 2017. We're currently working through our GLP biodistribution toxicology studies right now, and then hopes to have that filed by the end of the year. This indication has orphan drug designation and rare pediatric disease designation as well. We also have some plans to work in arthritis and other related conditions. These are in the early research stage. So today, as I mentioned, I'm going to focus on the first indication, but before I do, I'll talk a little bit about the platform. So these are autologous fibroblast cells from just our years of experience of working with these cell lines. They're collected from the patient from a simple biopsy punch. They're expanded in a GMP manufacturing suite, and at early passage, we are using a viral vector to deliver our genes of interest. So, for instance, in RDEB, it's a rather large gene. I'll talk about the protein in a moment. So we're using lentivirus to bring these genes to the cells. Their integrative nature and their large packing capacity were attractive for ECM proteins like this. So we're using that in early passage, and then we grow these cells up, and they're cryopreserved, and then they're delivered to the patient overnight after being prepped, and then they're administered locally to the areas of lesions and where we're targeting the product. So just moving into RDEB. So as I mentioned before, this is a rare blistering disorder. It affects children, also adults, but I'll talk a little bit about the impact on their health. One of the reasons we targeted the disease originally is it is monogenic in nature, so it is a culprit of this is a single protein type 7 collagen. And it forms a protein anchoring fibril that binds the 2 layers of skin together, and I'll show you an infographic shortly about sort of how the disease manifests. So what we've done is taking FCX007 is really reintroducing a synthetic version of that gene that was developed by us and in partnership with Entrexon, who we work with on our indications, and to deliver that back locally to the area via intradermal injection, and then try to make a functional change in that wounded skin, so create anchoring fibrils, have wound healing, and obviously want to make sure those wounds stay healed. So on top on this image gives you an idea about how RDEB affects patients. In our normal skin on the left, there's this sort of, you look at that green arcing structure going into the dermal layer, they're called anchoring fibrils, and it's almost like Velcro if you think about it. It's a hoop, that comes down and it binds into the lower ECM protein of your dermal layer, and then when that's a tight fit, then your skin stays attached. It's a mechanistic property. Unfortunately, in RDE patients, due to their mutation, they either create improperly formed collagen 7 or they're null, and their layers of skin tend to just float on top of one another, so any sort of lateral friction or damage to the area creates terrible blistering. And then the problem is they could heal, but then they re-wound, and then they're in this consistent either wounding cycle or have chronic wounds that can lead to a lot of terrible conditions. So it's progressive. They get a lot of contracture tissue. They're usually wheelchair-bound later in life. It actually has a very high mortality rate. Number one problem is squamous cell carcinoma and sepsis really affect these patients, and 76% of RDEB patients don't make it past their 30s. So they do have a very painful life, and it's a lot to manage. Not only is there a medical, obviously, issue with this disease, but there's also the care. And right now there are no approved therapies. It's mostly palliative. It's really managing wounding with bandaging and antibiotics. There's a lot of types of surgeries they have to get over the years, esophageal dilatations and finger separation surgeries. And there's an economic impact on families because they have to pay for all these supplies, and their wound changes can be daily, and it can be quite expensive, exceeding $10,000 per month, and not always reimbursable. Obviously, epidemiology-wise, this is a rare, what we would call an ultra-rare disorder, about 1,100 to 2,500 patients U.S. There is a broader disease called dystrophic EB. It includes a dominantly inherited version. They don't have the same mortality rate, but do still suffer from blistering conditions. So we're in the midst of a phase 1/2 trial now, and its primary objective is obviously to look at safety of this product. We're also looking secondarily at some wound healing endpoints and also pharmacology as well. And this trial is designed with 12 patients, 6 in phase 1, 6 in phase 2. We're currently enrolling adults in the trial. So, and then in the phase 1 portion, in the phase 2 portion, we can continue to enroll adults, so we can move straight through from the phase 1. And we're also petitioning FDA in phase 2 to allow pediatrics to enroll in the trial, which would be age 7 or older, so we can either have adults or pediatrics in the second portion. This is being run at Stanford University, and right now the status is we have 3 adult patients who are dosed, and we have manufacturing complete on a 4th patient pending dosing, and we just, as I mentioned last week, gave an interim data readout on our first 3 patients I mentioned. I'll talk a little bit about the data that we saw here. So this is a quick summary of the release. You know, we looked at 3 what we call NC1 positive adult patients. It's one of the cohorts in our phase 1. And we looked at them up to 12-week post-administration of FCX007. This was a single injection session. So in our history of injecting autologous fibroblasts, we've done successions of injections in areas where we would do it every 4 to 6 weeks, or some sort of therapy-based continuation of dosing. Here, you know, obviously due to the concerns of gene therapy, we started with a single injection session just to confirm that there's safety involved, and then go from there. The wounds retreated range from about 4.4 to 13.1 centimeters squared. From a safety standpoint, FCX was well tolerated through 12 weeks post-administration. We didn't see any SAEs or any product-related AEs. There was no RCL detected. If you work with lentivirus, we test not only the product but the patient's sera for any replicative competent lentivirus. We didn't see any of that. And we saw no low to no antibody response, which of course also didn't link to any sort of AEs. So we were very encouraged by the safety data. We also saw positive early trends in pharmacology and wound healing signals, so I'll talk about those in the next couple of slides. Based on this data set, our DSMB reviewed the data and concluded that safety and what we call potential benefit were established in those 3 patients, and then they allowed continuation of our trial. So now that we have this first initial injection session, we have some data. Obviously, it's early, but we're going to continue to follow these patients out, but we're going to be looking to implement some either dosing or some expression changes that we think will help benefit patients going down the road. I've described some things we have considered for the future, including repeat dosing or increased, or some things we can do for boosting expression. And we're going to continue to dose phase 1 adults in the 4th quarter of 2017. So starting with the wound healing endpoint that we captured during the trial, we did this through digital imaging, basically captures and tracing of wounds on patients, and obviously did that over their visits. We did that also by using skin tattoos and overlays to try to basically align ourselves to the wound so we can follow it on in future visits, post visits. These wounds were all monitored for a period of time before they were selected for dosing. And that period of time, we saw these wounds that were open for up to 8 months in our data set. So, obviously open wounds for quite some time. And then compared to the baseline measurements collected at day 0, so prior to administration of the product, we saw wound healing for just a single injection of FCX07 out what you see in the table here. So 5 of our 5 wounds, by the way, we dosed 5 wounds in these patients out of the 3 that were in there, so some had a couple of wounds. We saw that there was all 5 were greater than 75% healed at 4 weeks post-administration. And then at 12 weeks, we saw that 80% or 4 out of 5 were greater than 70% healed post-administration. We had noted on one of those 12-week wounds that the investigator had collected biopsies for pharmacology in the middle of the wound bed. So it is potential that there was some destabilization of that wound. We're obviously going to follow that up for investigation to see if that was the root cause for the one that dropped below our threshold there. So below is an image of one of our patients' results, and just to give you some orientation, the dotted line gives you the area of the treated wound. The sort of pen-colored, purple-colored dots are where the injections were performed, so you can see around the periphery and then crossing the wound bed. The 2 black dots on the left are the tattoos we give just to orient ourselves to the wound each time. And in this particular example, we could see that at 4 weeks, the solid lines trace the remaining wounding area, about 25% wounded left, and by 12 weeks in this particular example, this was covered by the time we reached the 12-week endpoint to 0% wounded. This particular wound was open for just under 8 months, maybe by a couple weeks. So, we were obviously encouraged by this result. It was originally a 10.3 centimeter square wound. So moving on to pharmacology. So in order to test for pharmacology, we have to take a biopsy punch of the treated area and draw out samples. So we are limited in our ability to sample these patients because if you over-biopsy the area, you can actually potentially destabilize the wound by not only removing cells, but just creating a physical damage to the area. So we did have a limited look at pharmacology from that standpoint, but we did conduct a number of tests in order to try to find some things that's going on under the skin. And what we had a result were variable signals across the population up to 12 weeks. We performed qPCR to look at vector DNA and mRNA present into the skin, electron microscopy to look for actual anchoring fibrils, and also immunofluorescence that we can use a human collagen 7 antibody to tag the actual protein in the cross sections. And we saw these signals throughout the population, including mRNA, DNA, variability through the area. We did not actually observe anchoring fibrils physically. It could be due to the low sample size. Could also be timing and protein expression. So we're going to continue to monitor anchoring fibrils and as we go to increase dose and expression. But we did see protein expression. Here's a sample we collected below. This is a cross-section of skin. Above is the epidermis, below is the dermis. And the arrows are pointing to what's called the basement membrane zones, so the layers between the skin. And that's where we want to see anchoring fibrils forming. So that's where we want to see collagen 7 actually being expressed and then homing to the area. So here we saw a nice result of linear expression of collagen 7 as it moves through that basement membrane zone in the area of the skin. And so this, again, is encouraging because the mechanism seems to be intact. In this one, obviously, we want to collect more samples and see that replicated more and more through the population. So, obviously, this has been a busy year for Fibrocell. We started in January being granted Fast Track designation. We moved through the injection series of our first 3 patients. Based on our protocol from a safety standpoint, we wanted to look at the first patient and then have our DSMB release that from a safety standpoint, take a look, and then we went on and injected 2 more patients. And then, of course, this interim update that we just put out last week. So we were happy to hit those milestones. And obviously for our 013 program, we were granted RPDD earlier this year and are targeting to have that into an IND by the 4th quarter of this year in 2017. In the 01, back to the 007 program, we are now, as I mentioned earlier, working on to dose additional patients in the adults in the phase 1, and obviously we can move on then into phase 2 in adults in the 4th quarter of this year. And then as I mentioned earlier, with benefit and safety, we'll petition the FDA then to allow us into the pediatric enrollment portion of the trial, which will be also follow on into the phase 2 portion, and we're targeting the 1st quarter of 2018 for that one as well. From financials, We had cash of $15.9 million at June 30, 2017, and we have funds on hand to fund our operations into the second quarter of 2018. Okay, well, thank you very much. Appreciate your time.